people can attend maybe.
Yes, yes, but it will be midnight for you.
Yeah.
But this will be recorded on YouTube, so many people can see this.
Oh,
Later, yeah.
Yes.
Good, good.
Yes.
So can I start, sir?
Yeah, first I will make co-host to Michael, sir. And I will make co-host to you also. Now you can start.
Yeah, okay, sir. Good evening, everyone. Welcome to the Microbiology Society India. We are conducting a lecture series from international speakers on biotechnology. So welcome to the lecture series. Today we have our speaker, Professor Michael San Francisco, Emeritus Professor and Dean, Texas Tech University. It's a privilege to have you, and I take the privilege to introduce a brief about Michael San Francisco Professor. During his 31 years at Texas Tech University, he taught classes in general microbiology and bacterial physiology to undergraduate and graduate students. He mentored 13 doctoral students, 10 MS degree students, and over 60 undergraduate researchers. He has over 50 referred publications, and his research was funded by National Science Foundation US, Department of Agriculture US, Department of Defense SBCom, and Office of the Vice President for Research. His scientific research and publications are primarily in the areas of molecular mechanisms of microbial drug resistance, atmospheric microbiology, and molecular biochemical analysis. Analysis of symbiotic and pathogenic interactions of microorganisms with their plant, wildlife, and human hosts. His research recently focused on the fungal pathogen Batrachochytrium dendrobatidis that has been identified as an agent in the global decline of amphibians. At the university, he served as interim chair of the Department of Biological Sciences, interim vice president for research, dean of the Honors College, and interim dean of the College of Arts and Science. So today, it's a really great to have the lecture, and I'll hand over to Professor Michael San Francisco to start the lecture.
Thank you very much.
Over to you, sir.
Thank you. Thank you very much, Dr. Sujita and Dr. Deshmukh, for this invitation. And I'm really happy also that other people have joined us, and Dr. Bhagyaraj is here because he was my inspiration in India. So thank you very much. So I'm going to share my screen.
Yes, sir.
Can you see my screen?
No, sir.
Oh, sorry. I'm sorry. One moment. Can you see it now? Can you see my screen?
It's visible, sir.
Okay, wonderful.
Yeah, you can start.
So I'm going to talk to you all about some of the work that we've done over the last 10 years or so on this fungal pathogen of amphibians, and it's commonly called as a chytrid, and the genus and species names are Batrachochytrium dendrobatidis. And so this is a fungus that was discovered about 35 years ago, and it has caused a devastating decline in amphibians worldwide. And so we got interested in it, and initially I was invited to do this work by a mathematician, actually, who was studying populations. And she asked me, because she knew I was also doing mycology, so she asked me to join her group. And so then we were funded from the National Science Foundation that initially funded this work. I just want to say at the beginning, all these students, Angela Moss, Joe Stekiniat, Shalika Silva, Amanda Starr, these are the 4 PhD students who did much of the research I'll be talking about today, besides a lot of undergraduates. So the first thing I want to talk about is the contributing factors that have brought about the decline of amphibians globally. And by amphibians, we mean broadly the frogs, toads, salamanders, newts, and those kinds of organisms, as well as a kind of a worm that lives underground. They're called caecilians. And so it's these are the major species that have been impacted by this particular fungus, Batrachochytrium dendrobatidis. From here on out, I will just say chytrid or I'll say BD. In the lab, we call it BD. So I'll say BD. So among the contributing factors, obviously, are habitat destruction, deforestation, and us building, as the human population grows, we know what happens to everything else around us gets destroyed. And then obviously secondary impacts of a lot of what we do, right? Our industry, our automotive use, etc., and the acceleration of the changing climate have also been thought to contribute to the decline of these amphibians. Also introduced species. There's a lot of invasive species all over the world that are now outcompeting with these organisms that live in their normal habitats. Some people have also proposed that ultraviolet B radiation has brought about some of this decline. Obviously, environmental contaminants, herbicides, and pesticides play a big role as those chemicals enter water systems. Amphibians have to spend part of their lives in water systems, so that's another issue. And overexploitation. So people actually have used frogs and different types of frogs in research and things like that. So in the past, so these are also, and people take them as pets, and so they get decreased in the environment. And then, of course, the infectious disease, and that's what my research is going to be. And the disease is called chytridiomycosis. So what happens in this? I'm missing a slide. So what we see here in a typical infected situation, we see that the organism, the fungus, once it infects the host, the frog appears to be lethargic. It loses its ability to move effectively and loses its ability to hop around as they normally do. The posture of the legs is wider than normal, and they lose their what is called the righting reflex. So I'll describe that later on in more detail. But basically, if you take a frog and you set it on its back, then it will normally be able to turn itself right over. But if it's been infected with this fungus, then it cannot move anymore. And so this is thought to be some neuromuscular issue, and I'll describe that later on with our research as well. Also hyperkeratosis. So the skin epidermal layer starts proliferating at an uncontrolled rate, and the frog begins to lose a lot of its skin on the surface. Also, because of this infection, there's an electrolyte imbalance within the animal, and it's thought to result in cardiac arrest and eventually death of the animals. So what I'll talk about today is in 6 different sections. One is how we began our study. So we asked the first question, how does the fungus even know that the frog is present in a given environment? Are there some specific molecules that the fungus can move towards? So we studied some of that. I'll talk about that. I also talk about how once the fungus finds its host, how does it adhere to the host? How does it establish itself? Can it bind to the mucus layer? Does it have a way of dealing with the antimicrobial peptides on the surface of the skin or the mucous membrane of the frog? Then how does the fungus penetrate? How does the fungus then impair the host? How did it prevent the host from defense? And how does it get dispersed in the environment in the absence of the host? So how does the fungus survive when there's no host around? And then we've also identified 2 model systems to study this fungus in the laboratory, so we don't have to use frogs to do this work. So briefly, I don't know if you can see my arrow, but in its motile initial stage, the fungus in panel A on the left represents a zoospore. So it's a swimming structure. And this is about a 10 micron diameter organism. It's called a zoospore, and it's motile. And the zoospore can, after some couple of days, can change or develop into a sporangium-like structure, which is a fruiting structure. It further develops, and then it is within it. If you look inside the sporangium, we see actually the formation of baby young zoospores. So now it's called a zoosporangium. Okay, and then the structure opens out, and about 1,000 or so zoospores come out from one zoosporangium. This is an important point to remember. Okay, each zoosporangium can generate about 1,000 zoospores, and the zoospore, this motile stage, is the infective stage. So the question was, how does a chytrid find its host? So what we did was we took hemocytometers, which is basically a glass slide with measured markings on it, so we could measure volume, and we took about 10 to the 7th zoospores in suspension, put them on one end of the slide. Here you can see on the bottom where it says zoospore suspension, and then we put a substrate, some kind of attractant molecule, on a disc at the other end, and we monitored over time for 45 minutes, 0 time, 45 minutes, 90 minutes, monitored movement of the zoospores towards the substrate disc. And so we did these experiments, and what we observed were, we studied a bunch of sugars, we studied some amino acids, we studied some proteins, and we found that substances like lactose, as you can see here in the orange over here, and glucose in green, were good sugar attractants for the fungus. We also found in the lower panel that casein, the milk protein casein, amino acid glycine, gelatin, hydrolysate, were also very good attractants for the fungus. And then we also studied, keratin and other glutamic acid, like cysteine and glutamic acid, as attractants. And you can see there's varying amounts of movement and chemotaxis towards these things, but basically we are able to show that the zoospore has chemotactic ability. It can detect chemicals in the environment and move towards them. At that time, we weren't sure exactly what it was being attracted to, but we just wanted to show through these studies that the fungus could actually be capable of chemotaxis. So once once the fungus makes contact with the host, the first thing it's going to encounter is the mucus layer. So the mucus layer is present in our GI tracts, in our gastrourinary tracts, the respiratory tract, and on the frog is on the surface of the frog. And so the question was, can the fungus move towards mucin? Mucin is a substrate we use in the lab. And sure enough, we did see positive movement towards mucin, as you can see here in the dark green panel, the dark green bars. Mucin is a high molecular weight, heavily glycosylated group of proteins. They're produced by epithelial tissues in most animals. And so we took agar plates with mucin impregnated into it, and we grew the fungus, as you can see here in the middle. So that's a fungal colony. Okay, and then we could stain it with a stain called Alcian blue, and you can see here that there's clearing around the fungus, and that shows that the fungus is capable of actually degrading mucin. So then we were interested in this, and we wondered about the capability of the fungus to produce peptidases and other proteases to degrade, the host and work in the host. And so during this period of time, the genome sequence of BD became available slowly, And so we were able to do some analyses, and we found that there were actually about 25 genes in this M36 domain of metalloproteases and about 29 genes in the subtilase protein group that were present in BD. Interestingly, these similar expansions, as they are called, are also found in human dermatophytic skin type fungi like Mycosporum and Trichophyton. So at this point in time, this chytrid, the BD, is not a human pathogen, but it has the capability to do a lot of things that human pathogens do. That's one thing to keep in mind. So one of the other things was we asked the question, can BD degrade elastin? Elastin provides, it's a protein that's found in the extracellular matrix, and it provides flexibility and elasticity to tissues. And so we used a Congo red elastin assay, and we were able to show that BD can degrade elastin. And you can see here in the blue bar that there's a high degree of elastase activity capable of the capability in this fungus. And it's a this elastolytic ability is a metalloprotease function that makes that happen. So that was very interesting to us. So what we did was we took some proteins, basically, like gelatin and casein, and we impregnated protein gels. And then as we separated the proteins in the gel and then we stained them later, you can see here the clear zones, these clear bands, indicators that these are the regions where there is a protease capable of, in this case, in the left panel, degrading gelatin in 2 places. And then in a casein zymogram, you can see this at three or four different places where the fungus is capable of degrading casein. And so those are where those proteins are in terms of size. And then we could then decide to go after them and identify them and purify them and things like that. I don't know if you can see this clearly, but we actually took antibody derived against Aspergillus fumigatus elastase, and we cross-reacted it, and asked the question, will it cross-react with the BD, elastolytic protein? And sure enough, it does. There's a very faint signal here where the arrow is. I don't know if you can see it, but there's a signal there. So the point is, though, that we were able to show that BD does have this protein degrading ability, and it might have a weak reactivity with the Aspergillus fumigatus elastolytic capability. So one of the interesting observations people have made is when a frog matures, when it grows from an egg into a tadpole, then into a young adult called a metamorph, and then into an adult frog, we can see actually that the organism, as it's growing, this little tadpole, and maturing, it can get infected. So the mouthparts of the frog, of the tadpole right here, you can see these dark regions in this picture. That's where the infection has occurred. But tadpole infections are not fatal. So a tadpole that gets infected may get a little sick, may not feed as effectively, but it is not lethal to the animal. So what happens is, as a tadpole matures and becomes a young adult, now the young adults become very susceptible to the fungus and they will die. So we asked the question, what's happening during this period? It turns out that during this period, there is a large surge of thyroid hormone that is taking place within the frog that causes reorganization of organs and this and that. And so we asked the question, does BD respond to thyroid hormone? And the short answer is yes. So we took a chemical called T3, which is a mimic of thyroid hormone. And we did quantitative PCR, and we looked at gene expression, and we found that in the presence of T3, there's a high almost 20-fold increase in the amount of subtilase protein, which is one of those proteases I mentioned earlier, okay, that is highly upregulated. So this initial work and the work on this whole topic was done by my student who came from Kerala, Joe Stekinyet. He's now a faculty member in Purdue. And so this is very interesting. We did not characterize the other proteins. We just studied this subtilase later. So then he decided to purify the protein, and I'm not going to go through the details in the interest of time, but there was he went through a lot of steps in purifying the protein. And then we took this partially purified protein, and we asked the question, what is the role of these proteins in the fungus when it is interacting with the frog? And the big question was, can it degrade antimicrobial peptides that are present on the surface of the frog? And what you see here in the right panel is a MALDI-TOF basically scan showing data of the intensity of the degradation of these antimicrobial peptides by this particular subtilase. And on the next slide, you see this more clearly. So if you look at this first panel on the top, you see PGLA is one of these model antimicrobial peptides. These are the amino acids in this, GMA, SK, etc. And you can see if you go down product 1, product 2, it gets shorter and shorter and shorter. Those are the products we were able to identify from the MALDI-TOF analysis, suggesting to us very clearly that this fungus is capable of actually degrading antimicrobial peptides that can be found on the surface of the frog. So we did this study for PGLA as well as for temperin. All of this work has been published, and that information is below. So a very nice piece of work was done by some people in Europe some time ago, and they showed these very, very interesting and informative pictures of, and if you look at the top left panel, you can see these are little zoospores that have begun to form on a light that's attached to the surface of the frog. And then as the zoospore matures into a sporangium on the surface, it forms a hypha. And then at the bottom of this hypha, over time, you see a formation of a new zoosporangium within the epithelium of the frog, embedded in the epithelium of the frog. And this zoosporangium can then rupture and then give rise to 1,000 zoospores per zoosporangium on average that can then be present within the animal. So now the zoospore is inside the host physically. I have this diagram below of the mucus barrier and pathogens that are often found in the gut lining of humans and animals. And so we were wondering, you know, since we have shown that BD can actually degrade mucin, and it can enter, and then once it can get free, quote unquote, okay, then can it produce its antimicrobial peptides? Can it produce its proteases? And can it produce other toxins that can impact the host and kill it? So that was probably one of our interesting places of interest. So one of the papers that we came across in 2010 was that there was a phylogenetic analysis done on different fungal groups, and it was shown, and from this, and you can see here in this yellow box that I've drawn around here, that some of the BD genes are fairly closely related to Aspergillus fumigatus genes that are actually capable of making this toxin called gliotoxin. It's an epipolythiodeoxypiperazine type of toxin. It's called ETP. So we said, well, since these genes are present here, we're going to do an analysis and see if we can actually identify them in our BD in the lab strains. And so in the left panel here, you can see we were able to identify a number of BD genes that were present. We're saying here, yes, 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 that were similar to proteins found in Aspergillus fumigatus that are capable of making this ETP type toxin. One important thing to note is this is a non-ribosomal peptide synthase. It's not, these these are peptides that are not made with ribosomes. It's called non-ribosomal peptide synthesis. Okay, and this is the pathway that was surmised to or shown to be present in Aspergillus. So we were very interested in this as well. But we wanted to see if BD has these particular genes, then are they upregulated under certain conditions or not? And so we did a study where we actually looked at 4 of these genes: the dipeptidase, the glutathione, S-transferase, the non-ribosomal peptide synthase, and the thyroidoxine reductase. And we grew BD in its regular liquid media that we used. Okay, and then when we added lactose to the medium, we were able to see this great increase in gene expression of these 4 genes we were studying. If you remember from my second slide or so, when I showed the data for chemotaxis, we showed that BD can move towards lactose. Here again, there seems to be a response to lactose in the medium, and these genes get upregulated. We examined on the right side, you can see the gene order for making this gliotoxin in Aspergillus. These are in color on the top. And then we analyzed what is present in BD below. And it appears that some of the core biosynthetic genes for gliotoxin production are also present in BD. But we wanted to go further. So this slide that I'm showing you represents about 5 years of hard work of one of my students. She just finished a PhD recently, Amanda Starr, and she also is a faculty member now in Virginia. But she was for the first time able to generate, so she was able to generate a very clear identification of this molecule called cyclophenylalanyl prolyl with 4 different isomers. And this has never been seen before by this fungus. And each of these represents this particular one of the isomers. And so as we began to study it, and of course we had to collaborate with chemists and people at the Biotechnology Center at the university, she identified 113 different compounds, and these compounds are the ones we decided to focus on. Among the reasons we did that was because the fungus, this particular protein is, this particular novel metabolite is postulated to be important in antibacterial and antifungal activity. It's supposed to inactivate the cholera toxin. It's supposed to play a role in auxin-related gene expression in plants, as well as in quorum sensing. But very interestingly, it has also been postulated to inhibit calcium channels and play a role in immune suppression. Now, if you remember from my early slide when I said that the frogs that are infected die of electrolyte imbalance and cardiac arrest, so we were wondering whether this particular molecule can play such a role, in that aspect of the pathogenic, progression, as it were. So here's a little, this is a hypothetical thing that my student had put together, but you see, ordinarily, what is called the writing reflex, a healthy animal, which is on its back, okay, will respond to perhaps calcium and some neuromuscular activation, and it causes the animal to turn around in a proper way. So now it's back again. If these suites of molecules are present, then one or more of these isomers of cyclophenylalanyl prolyl, okay, you see that the frog is on its back, then there is now no activation or impairment of calcium channels, and therefore there is no activation of neuromuscular transmittance, and therefore now the frog cannot turn over. So these are the classic symptoms that we see in the frog. So it's conceivable. We have not actually tested this hypothesis, but it's conceivable that this may be happening in a super infection of the frog. So we decided to look at the alternate models for studying this particular pathogen, and we used a wax moth larvae. So you have to pay close attention to the figure here. So when there's no infection, the wax moth larva is, look at this, it looks pretty clear, okay? If there's a small light infection, then it turns light gray. You can see this very slightly. If there's a next level infection, you can see a black stripe down the middle. Then in a higher level infection, you could see it's dark gray with a black stripe. And if it turns black, then the wax moth larva is dead. This scheme has been set up for studying other fungi in this host organism. So we wanted to see how does it work with our BD. So if you look at the left panel, you can see a graph here where on the top horizontal line represents a phosphate buffered saline control. And then the lower line where you see this health index score, which is what these colors are, keeps going down if we infect the wax moth larva with BD, okay? If you look at the right upper panel, now you see a culture supernatant of BD. Again, this is the control on the top. It remains, the wax moth larva are fine. But if you infect them with this culture supernatant, now suddenly we see a great decline and a rapid decline in the health index score, suggesting that this chemical may play a major role, you know, obviously in inhibiting this organism, but maybe also BD. Then we tested the individual 4 isomers, the LL isomer, the LD isomer, the DL isomer, and the DD isomer. And you can see that 3 of them, the LD, the DL, and the DD isomers showed some good activity in impairing the health of wax moth larva. The only reason why we couldn't go further than like 60 hours is because these larvae mature and they form a cocoon. And so then it becomes hard to go beyond that point. So in any case, so this study also, all this work has been published, as you can see. So we also showed much earlier, about 10 years ago, that BD can infect nematodes like C. elegans. And we did a study, and this work was done by an undergraduate in my lab, Edwin Shepard. And we infected the slides of cultures of the nematode with BD. And then we used propydium iodide, which is a dye that can then penetrate when it's an unhealthy animal and bind to DNA. And we did actually some nice pictures here. So you can see here C. elegans in the bottom panel, and you can see the BD zoospores around the one place in the animal. And then because these particular worms' bodies are under turgor pressure, once the skin is ruptured, then everything comes out like a broken pipe. And so you can see the whole inside of the animal is coming out now. So anyway, so it's very interesting as well. So first part summary, you can see that BD can seek out its host by positive chemotaxis, can degrade mucin, it produces antimicrobial peptides like subtilicin, it can degrade the host epidermis perhaps with the elastolytic metalloprotease, and has the ability to produce toxin or toxins related to Aspergillus fumigatus. So the question is now, how does it survive without the host? Here's this, again, the same summary of BD with the zoospore maturing into a sporangium. So my student Shalika Silva did these studies using electron microscopy, and she was able to show that we see these biofilms. We call them microaggregates, but effectively they're biofilm-like structures where we see aggregates of sporangia that form. And if you look closely at them, you can see that there are these little zones where they appear to be connected. So we did a series of analyses of all of this, and we were able to show that BD does have the ability to form a monolayer of associated cells, and the extracellular matrix, which was that material joining all the different sporangia together, has all the sugar characteristics that are found in ordinarily biofilms. Furthermore, if we compared if you look at these three colors here, the green, the red, and the blue, the red in the middle is the monolayer cells, associated cells. The far left is the sporangia, and then the far right are the motile zoospores. And we did RNA expression analysis and studied all the genes that are expressed under each of these different stages of the organism. And you can see just by virtue of the color that there are some big differences, as well as some similarities. So there are more similarities between the sporangia and the monolayer than with the zoospore, which is in green. And we didn't go into a detailed analysis of this. I'm not going to talk about this, but we have all of those data in the paper that she just published in 2019. So it just goes to show there's a differential gene expression between each of these three forms. So some of the important and interesting genes that we identified were there's multi-drug associated genes, genes associated in biosynthesis of amino acids. The alkyl hydroxyperoxidase reductase gene is important in oxidative stress prevention. Phenazine biosynthesis was a bit of a surprise. Phenazines are produced by organisms like Pseudomonas aeruginosa. And so we don't exactly know what role it has in BD. And HSP70 that you might have noted before when we were studying the subtilase story also plays a role as a chaperone in protein folding. So there are a lot of things that still need to be understood here. So we had another study where I had lots of undergraduates in my lab we looked at a series of chemicals that we normally consume, like turmeric, garlic and ginger, and so wondered whether extracts from these different plants would be of value in treating preventing the growth of the zoospores in the lab, in vitro. And the answer is yes. So we were able to show that actually curcumin, which comes from turmeric, is effective at some relatively high levels. As you can see in this graph, I'm not going to go into every detail. You can look at this later. And then garlic also is very effective, as is gingerol. And we had to use different solvents. So curcumin and gingerol were used in DMSO as solvents, and allicin was used in just water or PBS. Furthermore, we were able to show that curcumin, gingerol, and allicin have different effects against the zoospores in the black bars, or against the sporangia, or against the monolayer associated biofilm cells. Okay, so you can see that different chemicals will have different impacts if you're trying to treat them. Many times you say, why are these studies important? Sometimes when people go into the forest and they find a mating pair of male and female frogs that have a low level of infection, and they want to bring them out and keep them in a veterinary environment, like a hospital, so they can reproduce in captivity, you may be able to treat these organisms with these chemicals that are quote unquote non-toxic at low levels, on the animal, but get rid of the fungus with them. We have not tested that yet, but that's something that's open to someone to do. So anyway, so in conclusion, we've shown that BD can establish itself on the host and degrade some of the host surfaces. It may be able to produce toxins that play a role in the lethal aspect of the infection. It also can infect alternate hosts in the environment, and can survive in aquatic environments, by forming these microaggregates, not unlike biofilms. So I think I'll stop there. These are my students who did most of the work. So each of them, that's Angela Moss, that's Joe Stecchinier, that's Shalika Silva, that's Lisa Matz, and Amanda Starr is down here. And so these are all some other undergraduates, Priyanka Maiti. So a lot of students in my time, over, 60, 70 students have done work in my lab, but I'm very grateful to all their work. So, and I'll be happy to answer any questions. Thank you very much.
Thank you very much, sir. On behalf of Microbiology Society India, I thank you very much for sparing your valuable time and giving a very informative lecture on BD amphibian pathogen survival patterns. And now the session is open for any questions. If you have any questions, please ask Professor Michael, sir.
Yeah, if you put it in the chat, there's some questions.
Yeah, there
There's
are
one that's, sorry. There's a couple of questions. One says, mucin degradation harmful among gut microflora. Well, mucin degradation, yes, is very useful for gut microflora to be able to get out of the mucus layer and then get into what are the intestinal crypts and then cause invasion. So yeah, so mucin degradation is useful for pathogens from that perspective. And how is lactose utilization relevant to chemotaxis? Oh, we were just interested in putting different sugars out and asked the question, does it move towards these things? So we at the time did not realize lactose was directly relevant, but we did show that it was a stimulant for gene expression in many things. So we don't know where the lactose is coming from or anything, but there may be some other sugars that are produced or in the environment around the frog that could play a role. I don't know that answer. Which media was used for growth of the chytrids? So it's called H broth. It's not unlike L broth, Luria medium. If you read our papers and the references are present there, you can find the exact composition of the media. So at least for BD, it grows very well.
Yeah, thank you very much, sir. And if anyone want to check back the lecture, they can check back in our YouTube channel. It is live streamed in YouTube channel. And so if there are no questions, then we'll conclude that session with this.
Yeah, Dr. Michael, I have a question. Like, can I have one question, ma'am, please?
Sure,
please carry on.
Because, the antibiotic resistance is increasing day by day, right? So, sir, Michael, sir, do you have any area which you are working to combat with the resistance pattern or resistant things? Because, I saw many newses about the drug resistance. So are you working on any area where we can just reduce this antibiotic resistance against fungals, against bacteria and other? Because many people is going to be like in a problem, like in a nearby future. So do you have any I was looking somebody who's working on synthetically, can we prepare some peptide based or something thing so that can in future they can fight with the multiple drug resistant things or something like that. So do you have any idea, something you can do just?
Yeah. So this is a very broad topic, but there's a lot of work going on in antibiotic resistance and ways to get around that, maybe if Dr. Sujita could give you my email or something, then I can send you some papers or something, that I can respond there that way better, maybe. Yeah, there's a lot of work going on in antimicrobial resistance. We've written one review article long ago, but maybe that'll be a good starting place at least. So yeah.
So sir, can we because I'm working here in Chandigarh University in Punjab, so can we just collaborate on some research?
Well, I have retired now, so I'm not actively doing research, but of course I intellectually collaborate with people.
Yeah, you can just mentor us that what should be done, so it's the exchange of ideas. We can go ahead with ideas so that we can implement here, and from there you can just mentor us that okay, in this way we can find out new molecules, or anything can be done, sir. We can try, because science we can exchange the things, then we can go ahead with this thing. If you would like to do, then I would like to also invite you for online any guest lecture in Chandigarh University as well, because I'm working there. So I just want to that you can interact with the students. We are medical students there, and these medical lab technology, microbiology, and we have like optometry student like working on the eye infection and all those things. So these things are increasing day by day. So we are scared that what will be if any pandemic situation like COVID or any outbreak can happen. Like if drug resistance will be there, would be there, then what will be the next? Everybody is scared because in scientific community we all scared that anything can happen at any time. So
Great.
thank you very
Yeah.
nice. You presented well, very nice. You have very good I impressed with your sir presentation. Thank you very much from my heart. So thank you very much,
you.
sir, for your presentation.
Thank you. I appreciate it. Yes. Thank you.
Yeah, thank you. Thank you very much, sir. Hopefully, we'll have more and more lecture from your side. So I wish for that one. Yeah.
All right. But I appreciate the invitation very much, Dr. Sujita, and thank you for
Thank
your
you.
kindness.
Thank you. Thank you very much, sir.
Thank you all.
Thank you,
Appreciate
Bala.
everything.
Yeah, thank
Okay.
you.
Have a good night. Okay. Bye-bye.
Good night.